Today's a short talk, 5 minutes, so I hope I can get everything in. But my laboratory is at Northwestern University in the Department of Pharmacology. And what we're really interested in is studying the developmental trajectory from the prenatal period and the early postnatal period for various ion channel diseases, including KCNT1. For summary of what I'll be telling you about, we have two recent studies. The first one in collaboration on the left with Tim Yu and Len Kasmarek, among others, which is a more clinically based study looking at an ASO treatment that was published this past month. And I won't be discussing any of the details of that study, but it's worth mentioning that the ASOs used in that study are the ones that we, in fact, are using in our study, where we're trying to target prenatally various types of tissues to look at the activation. So any specifics you would like associated with this data, it'd be worth looking into the papers, and I'm also happy to take questions offline. So the paper I'll be discussing, as I mentioned, is largely based in stem cells and various in vitro culture models, and we, at patient-derived stem cells, we're able to identify cell types in the early developing brain as well as the postnatal brain, where we can look at what cells are expressing KCNT1, including the inhibitory populations. And then if we apply this ASO, how we're able to differentially modulate excitatory versus inhibitory neurons. So in very brief description of the data, we were able to take the stem cells from an individual with the R474H variation and turn these either into excitatory neurons at the bottom or inhibitory neurons at the top. And one of the key takeaways that I want to show everyone here is that, in inhibitory neurons versus excitatory neurons, it appears that the cells are responding differentially to this ASO treatment. One of the key features that we notice is that when you use an ASO, which is the purple traces, and you elicit an action potential in excitatory neurons, we get a marked increase in the after hyperpolarization in the excitatory neurons at the bottom. But in the inhibitory neurons up here at the top, you can see we don't see the same sort of activation kinetics. Similarly, if we drive the cells harder and make them fire more action potential, so really push the systems, we can see that we get different effects of the ASO to recover high frequency firing in our ASO treated condition, while in the in the excitatory neurons, we see a bidirectional effect where at low frequencies, stimulations or activations, we get an inhibition of firing, and at higher frequencies, we get kind of an increase in the firing capabilities. And this is all, just to say further that when we're using these stem cell derived systems and we're differentiating them into neurons, either excitatory or inhibitory, we might be getting differential effects that are worth looking, more closely in the way we're performing these models. But as I mentioned at the beginning, one of the key drivers of the lab is what is the earliest onset and manifestation of ion channel genes? And for us, we're particularly interested in the prenatal period. So this would be a time during development when the brain is forming, there are cells dividing, they're migrating, there's cell death during this period of time during gestation. You'll see this week's gestation. We are typically working from the period of 15 weeks gestation through 23 weeks gestation. And one of the first questions we really wanted to ask is, well, when is KCNT1 most abundant during this period in time, and how is it functioning? Along the bottom here, you can see this would be human KCNT1 across this developmental time window. So the post-conception weeks, and then birth would be somewhere around right here. And you could see at the level of RNA, we have an increase in expression around early mid-gestation, 16 weeks to 23. So to go a step further, we're able to work with our colleagues in pathology to collect primary fetal tissue to take this and look at and evaluate the electrophysiological features of these neurons. What we wanted to identify is, where is KCNT1 in the developing brain? Is it in excitatory cells? Where is it distributed along the cortical column? And can we modulate this as a potential therapeutic target as an early intervention? So up here on the top, you can see this would be a piece of fixed primary fetal cortical tissue. We can localize KCNT1 to these neurons in the outer region of the cortical plate. And the first experiment we performed was, well, is there a sodium-sensitive potassium current? And the answer is yes. There is a functional potassium KCNT1 signal in the mid-gestation cortex. Next thing we wanted to know is, can we modulate this KCNT1 current in these neurons? And using the ASO that I previously described from the stem cell-based studies, we were able to target these receptors or this RNA in these prenatal neurons to decrease the amount of protein expression in these cells. And finally, if we look at electrophysiological features or neurophysiology, where we're seeing action potential firing in these prenatal neurons, we can change the way that they are firing action potentials or their excitation profile in terms of how they respond to this antisense oligonucleotide. Okay, so I know I went quite quickly through that because it's a very short talk, but one of the key takeaways I really want to reiterate is that we've studied various cell types in the lab. This includes stem cell-derived systems, which are IGABA and iNeuron here, and they respond differently to the antisense oligonucleotides. But also, when we take native tissue from humans, and we treat them with this ASO, we do see a different response profile from those cells. So when we're considering our biomarkers and our treatments and dosing, I get the sense that it's going to require a lot of detail in terms of understanding how inhibition and excitation is occurring in these circuits, because as we all know, there are many different types of neurons in the brain. And if the ASO is operating differently in each one of them to change excitation, we should be thinking more about the outputs of the circuit. Once again, thank you for your time. I'm happy to take questions. A lot of people were involved in the two different components of the study, including our colleagues at Boston Children's, Tim Yu, and at Yale. And thanks again.
I have some questions. That was really nice, Richard. Thank you. So I'm really wondered about splice isoforms. It's something we're getting much more interested in, but splice isoforms for other ion channels change quite markedly during development. And as you know, there are multiple splice isoforms for KCNT1. I'm just wondering if it's possible to determine if different stages, for example, the onset of myelination or earlier stages, are associated with changes in splice isoforms. That's one comment. The other comment is that, we've been having fun, I think like you have, looking at the Allen database information on where KCNT1 is expressed. And it's extraordinary how variable that is. So, for example, interneurons in the hippocampus, the paravalvulin, don't have KCNT1. Auditory system, those same interneurons have a lot of KCNT1. Frontal cortex, a lot in the pyramidal cells, but not in the interneurons. So that's going to be really interesting to figure out at a circuit level.
Yeah, certainly. The developing brain has a lot of changes in neurophysiology. Also, in the prenatal brain, there are very limited action potentials. So the KCNT1 could have non-canonical functions in the sense of not contributing to excitation in the way we think about it. It could be, what are its binding partners? How is it localizing with sodium channels? How is it activated, differentially during these periods of time? So it's certainly a very exciting time to be studying, with the types of technologies we have right now for single cell analysis.
What do you think is the phenotype of a KCNT1 knockout in humans? Is it embryonic lethal?
For a heterozygous?
Homozygous knockout.
I don't know if there are homozygous knockouts.
No, that's why I'm asking. There aren't that I know of.
So So by that aspect, I would believe that it's the KCNT1 gene is essential to the developmental process, and therefore it is required for machinery during this prenatal period. So I don't know what its function is yet. We're trying to get closer to that with our prenatal studies, where we're overexpressing in cell type. But what I will say, and it's worth noting, is unlike the lab spends a lot of time thinking about dividing cells and how they're growing during this prenatal period. And traditionally, we've looked at ion channels that result in brain malformation. So brains that don't fold properly, or things that structurally change the cortex. And those ion channels generally are in the cortical plate, so where your neurons are, but they're also expressed in these some of these dividing cells. And KCNT1 does not appear to be a part of that group in the sense that it is not expressed in these dividing cells or these early neuroblasts the way we see with some of these other very severe early ion channel diseases. So there might be some differences in the way that it's operating compared to some of the other ion channels we study in the field, like SCN3A, GRIN2B, ATP1A3.
Go ahead, Carlos.
Hello, I'm Carlos Fraga from Serbia. First, thanks for the talk. I really liked it. I wanted to ask you if you have seen whether the expression of KCNT1 is particularly high or low within any specific cortical regions, and if you have looked at the subcortical regions as well, or maybe this data is not available. Just wanted to ask you that.
We have not taken deep dives. So the Allen data set, you can go and take a look at. There's all these different regions, and as Len alluded to, that it can be quite noisy in the sense of, what does the RNA mean at the end of the day? Also, I mean, we see sometimes RNA be 3 times as high, and the protein doesn't appear to have 3 times more abundance or operating to control the cells in 3 times more capacity. So I don't have a great answer to differential KCNT1 enrichments across different brain regions, particularly in development. We focused exclusively on the developing brain, and one of the enrichments that we do see is this enrichment to the subplate, and there's more detail within the paper. This would be like your earliest, deepest neurons, and then we see some enrichments in various cell types out here. In the postnatal brain, inhibitory neurons, obviously, we see parvalbumin, somatostatin have some significant enrichments, but this time during the prenatal is quite dynamic, and things change pretty quick. So, yeah.
Thank you.
Okay, let's do one more question from Lauren, and then we're going to move on to our next speaker. Go ahead, Lauren.
Hi, Lauren Black from Charles River Labs. I just wanted to know, when we struggle a lot with trying to replace animals with cellular models, which I am, having worked at NIH on cellular models, I'm quite pro. But I was a little bit worried about the fact that you get differential effects at low frequency and high frequency stimulation. And which one do you think is more physiologic, and how do you know?
Great question. We struggle with this often. It's a little bit of a scary thought to think that our primary neurons, which we think are most like excitatory neurons, so like the eye neurons, might be responding differentially. So a native piece of tissue could be responding differently to our best stem cell derived neuronal model systems in terms of selecting biomarkers. To your point here, about what is the relevant physiological range, KCNT1 is known for many years to be involved in high frequency firing, coding, salience, these sort of things where cells, rely on repetitive bursting. So I would go as far to say that these high frequency patterns do matter. These high stimulations and seeing a cell the way it can handle repetitive action potentials and firing frequency. So that being said, getting an increased AHP is a bit phenomenological in the sense of what we think about in terms of KCNT1 more by knocking down or removing KCNT1. One would think that you would reduce the amount of your AHP because of the potassium, the way the potassium exits the cell. So there's a lot of nuance in terms of, or things in terms of the way these cell models are built and potentially the way they're compensating. But at the end of the day, this is similar to the SCN8A story where there might be something about KCNT1 and modulating it that allows these cells to perform, these high frequency firing patterns here in the disease case. That is otherwise not possible. So